going to start with chapter 3 and then move on to 17a. Okay, this chapter, chapter 3, is about movement across membranes. So when we talk about membranous tissues, you can have what we mean by membranous tissue is something you see with the unaided eye. So we're talking about a tissue that you peel away. And a good example of that is the pericardial membrane or pericardium. So notice in green here should be the blank that you have to fill in. Epithelial tissues, 1 to many cells thick. So this is a little bit smaller, but these are both examples of membranous tissues made up of many, many cells. The other example, or the other meaning of membrane, is the cell membrane. And another name for that is the plasma lemon. This encloses cells. So notice that here we have a cell membrane at the bottom around each cell, but you also have a macroscopic membranous tissue around the heart. Okay, this is an image, I believe, from your text showing you the different molecules that you would find in cell membrane. There's phospholipid bilayer, channel proteins allows things in and out, integral membrane protein, which stabilizes or could act as an enzyme, and then glycoprotein is a protein with a carbohydrate attached. One of the things you should know about this is when there's a carbohydrate attached, this thing here is part of the glycocalyx, and this is what the body uses, or the immune system uses, to determine if a cell is part of you or an invader. Okay, membrane proteins that bind with molecules. Now, what we're looking at here is a receptor, often made out of protein, and the ligand binds to it. A good example of that would be a hormone binding to a receptor, causing an event in the cell. So this is a ligand receptor complex, triggers an event in the cell, often a hormone. Membrane-associated enzymes may catalyze or cause to happen external reactions such as digestion in small cells. For example, there are enzymes on the surface of your cells, of your digestive tract, that help you finish the digestion process of your food. Internal reactions can be in terms of transferring external signals to the cytoplasm, and that's what this figure is showing here. Receptors bind specific ligands. Notice that they have the exact same shape. If the receptor is not the same shape as the ligand, it will not bind and nothing will happen. So a good example of a ligand is a hormone. Cell recognition molecules we will be looking at later, and I showed you a figure earlier on. Transporter proteins, moving substances across membranes. So a good example of that is a channel protein. I showed you an image of that earlier in the presentation. If it's an open channel protein, it creates a water-filled passageway to the ECF, which is extracellular fluid, and it can vary in sizes. So larger channel proteins allow larger molecules through, and smaller channel proteins only allow the smallest molecules through. If it's gated, it's still a channel protein, but it can be open or closed. It's usually closed, but can open to allow passage of certain molecules. For example, sodium and potassium, we'll be talking about later, have gated channel proteins that allow those molecules through at particular times when they need to go. They're less selective, which means that anything small or smaller than the whole can pass, usually. And they're often called pores. And we have pores in our skin, but nuclear pores are found in the surface of the nuclear envelope that allows materials in and out, especially messenger RNA, to get out of the nucleus to do protein synthesis. Now, carrier proteins are different. They bind directly to the substrate and only open to one side of the membrane at a time. And the way to think of this is kind of like a revolving door or a turnstile. The idea is that one person goes into the turnstile, moves through, and then another person moves through. It's less of a doorway, more like a turnstile. So it's slower, but it's more specific. So the downside, it doesn't move things as quickly, but the upside is it only moves the things that are supposed to be moved and nothing else. Okay, here's that same image that I wanted to show you. This glycocalyx, and it's made up of a combination of primarily glycoproteins, but also glycolipids. Notice that they're sticking out from the surface so that white blood cells involved in cell recognition can check to see whether or not this is you or an invader. So it's used in cell recognition, cell stability, and the immune response. It's composed of both glycolipids and glycoproteins, and you see them here in this image. I believe this image is from your text. Okay, body fluid compartments. Now, if it's inside an individual cell, it's called ICF, or intracellular fluid. It doesn't say F here, but it stands for fluid. The F is for fluid. So intracellular fluid is ICF, and that is synonymous or the same thing as the cytoplasm. In extracellular fluid, it's any fluid outside of the cell. So there's two different compartments that make up extracellular fluid. It can be interstitial, and this is the fluid you find between cells, but not inside of blood. So there is fluid between the cells, and this fluid ends up in your lymphatic capillaries and lymphatic ducts and your lymph nodes, and it becomes what is called lymph or lymphatic fluid. Plasma is the liquid portion of blood. This is also part of extracellular fluid. Okay, now one of the essays I'm going to be asking you about is this thing called Fick's law of diffusion. So you really want to take detailed notes here on how it works. Now, what we're going to be talking about is how the body wants to maximize diffusion by affecting this equation. Now, I know when you hear something that looks like math, your eyes roll to the back of your head and you stop listening. But I'm going to explain this in detail after I do the notes, and I'm going to explain to you what you need to say in your essay when I ask you, tell me as much as you can about Fick's law. Okay, first of all, we're going to talk about diffusion. Diffusion stops at equilibrium. What that means is when there's equal amounts of material on both sides of a membrane, it's not going to continue to change. It's going to stay the same when it's equal. Now, particles are going to be moving across the membrane, but equal amounts in and out at equilibrium, which means even amounts on both sides. Now, what are some factors that affect how fast things diffuse across the membrane? Well, we can talk about surface area. That's down here in the purple. Temperature, which is not part of this equation. Distance, which is here in thickness of membrane. Please note, distance and thickness of membrane are the same thing. It's the distance through which materials have to move across the membrane. Lipid solubility is over here in this part of membrane resistance, and size of molecule diffusing is also part of membrane resistance. Okay, so this is not in your text, so you need to listen to what I have to say. Take detailed notes. Now, the nice thing is, you guys are watching this on a recording, push pause, take notes. Please do not go out on the internet and try to figure out how to discuss Fick's law of diffusion by grabbing something that you see there, because that will be worth nothing. Listen and take detailed notes. Okay. What we're talking about with Fick's law is a fraction. Now, if you remember, there's numerator on top and denominator on the bottom. So what is going to happen with the body is it wants to make everything on the bottom small and everything on the top large. For example, if you look at a fraction like 1/3 and then compare it to 1/4, the number on the bottom, 1/4, is bigger than the number 1/3. The question you have to ask yourself is, what is the larger overall amount? What is more water, 1/3 of a cup or 1/4 of a cup? If you know anything about measurements, you know that 1/3 of a cup is more than 1/4 of a cup, because the larger the number on the bottom, the smaller the overall value. So for example, 1/2 is more than 1/5, because the big number on bottom means you have to cut it that many more times. So what the body's trying to do is make the stuff in pink here as small as possible and the stuff on top as large as possible. For example, 1/3 or 2/3, which is more? Well, you know that 2/3 is twice as much as 1/3. So having the larger numbers on the top means that you're going to have more overall amount. And in this case, we're trying to get the rate of diffusion, how fast things diffuse. And a perfect example of that is oxygen getting from air of your lungs into the blood. You want that to happen quickly. You also want food to diffuse quickly from your digestive tract, the lumen, where the food is, into your blood so you can transport it through your body. So let's talk about these various things I'm going to talk about. You guys listen. Okay, so let's talk about the stuff on top first in purple. Available surface area. Now, if you've ever heard the expression, the shortest distance between 2 points is a straight line, that means you're going to have a very small amount of surface if it's flat. Flat is low surface. But instead of making it flat, what if we make it curved, like a wavy line? Since that is not a straight line, it is a much longer line and a much greater surface area. So what the body does in places like the lungs and digestive tract, anywhere where you want a lot of diffusion happening quickly, instead of making it flat, it makes it curvy. So if you look on the inside of the lungs, you see the alveoli are curved like the inside of a grape cluster. And the surface of your small intestine has these finger-like projections. And each of those finger-like projections has little projections sticking off of that, which vastly increases the surface area. So when I ask, what does the body do to maximize diffusion anatomically and physiologically? One of the things it does anatomically is make sure that where diffusion has to be quick, there's a large surface area. It does that by making it folded. Lots of folds and curved surfaces increase the surface area. That increases the rate of diffusion. And in that way, you get more food into your blood and more oxygen into your blood. Okay, that's surface area, or A. Okay, the next thing that we're going to increase here is concentration gradient. This is a physiological modification that your body does. Now, what do we mean by concentration gradient? Well, the difference between inside and outside is the gradient. So let's say you're going down the grade is a hill, and there's a big difference between the top of the hill and the bottom. So the gradient means what is the difference in concentration between the inside and the outside. And the greater the difference, the faster diffusion happens, because more stuff is going to go that way and comes back the other way, because there's a whole bunch more over here than over there. And so it goes that way faster. And as soon as it gets closer to equilibrium, then as one particle goes this way, another one's going to go the other way. So noticeable diffusion slows down. So what does the body do physiologically to increase concentration gradient? Well, let's go back to the lungs. Well, you're not going to let the blood get to equilibrium, because when it does, you're not going to be picking up any more oxygen. So what the body does physiologically is, as soon as it picks up enough oxygen in the lungs, it moves it out and brings in more deoxygenated blood, so that there's always less oxygen in the blood than is in the lungs. Because as soon as it got saturated with blood, you move it out of the way and bring in more that is unsaturated. So that what that means is there's always going to be a gradient from the air of your lungs into your blood, because you move the oxygenated blood out, bring in more deoxygenated blood. That's a physiological adaptation to increasing the rate of diffusion by making sure that the concentration gradient stays as high as possible. Okay, that's the numerator. We want this to be large. Curved surface, concentration gradient by moving the oxygenated blood out of the way. Now, let's go to the bottom, membrane resistance. Now, this is a little bit weird, but membrane resistance is shown by these two things. Now, what I'm going to ask you guys to do, I know this is a little bit confusing, but what I want you to do is put lipid solubility up in the numerator and molecular size in the denominator. So lipid solubility goes up here with the purple, and molecular size goes down with the pink. So we're going to add lipid solubility here, and we're going to take out membrane resistance and put a molecular size on the bottom. Okay, so let's talk about these. Lipid solubility. That means the ability for it to go through lipid. Now, why is that important? Well, notice that the center of the membrane is all oily, or lipids. If something can diffuse right through lipids, it can get right through this oily core and make its way through. So if it has lipid solubility, that increases its rate of diffusion. So lipid solubility goes up on top. Molecular size is the opposite. Bigger things move more slowly than smaller things. How long does it take a cat to get through a door versus an elephant? It takes the elephant a lot longer, because it's a big object that needs to start moving slowly and eventually goes. The cat can go through very quickly because it's a small, much smaller object. So when we're talking about molecules, large molecules are going to diffuse slowly. So the bigger the molecule, the less quickly it diffuses. Now, the body can't do anything about this, because you have to diffuse what you have to diffuse. But the molecules primarily that are going to be diffusing are in the lungs are going to be oxygen and CO2, fairly small molecules. But in the digestive tract, you can either move proteins, which are really big, or amino acids, which are much smaller. So what the body does is it breaks it into amino acids, making it a smaller molecule, and therefore allowing it to come in more quickly. That's a physiological response. Break the large molecules into smaller ones, and you can get them through quickly into the blood and use it as nutrition. Okay, so lipid solubility goes on top, molecular size goes on the bottom. Now, on the test, I'm going to actually ask you to write this out. And if you don't, you won't get it. So how do you write this out? Well, you can, on one line of your answer, you're going to write these words: available surface area, concentration gradient, lipid solubility, all on one line with a little X for times. Then you're going to go to the next line, put a dotted line or an underline. Then the third line, you put molecular size in place of membrane resistance and thickness of the membrane. That way, I know that you know what the equation says. So the last thing that we're going to do is look at thickness of membrane. Now, if you want to get people from one side of a structure to the inside of the structure, do you have them go through a long hallway, or do you have them go through a fairly thin door? The answer is go through the thin door. Now you're from outside to inside quickly. So by having this membrane be thin, it gives less time that it takes to get through it. So what we're going to see in the lungs, again, and in the digestive tract is very, very thin membranes. So the distance from the outside to the inside is minimized. So when we say thickness of membrane, if you want diffusion to happen quickly, if you want plenty of oxygen, you want plenty of food, you make this very thin. That's why when you look at the inside of the lungs, the surface of the inside of the lungs, you're going to see a very, very thin membrane, and the same with the digestive tract. That's another anatomical adaptation, is by making this membrane very, very thin. That increases how fast things go. Now notice it says temperature. Pretty much we keep temperature stable, but in a test tube, if you want things to diffuse more quickly, you make it warmer. And that's part of the reason why we're warm inside, is to increase the speed of all these chemical reactions. Okay? Now, you guys aren't here to ask me questions because nobody came today. That's okay. But if you watch this tonight and you have questions, if you do come to lab tomorrow, be sure to ask me questions about Fick's law, and I will answer them for you. But I can't, you guys didn't come for this. All right, so we're moving on from Fick's law. Hopefully you guys can get plenty in there. Yes, you can look on the internet, but do not copy anything you find on the website or in your text. I don't even think this is in your text, but take good notes. You should be good to go. All right, facilitated diffusion. What does facilitated diffusion mean? It means that even though there's a concentration gradient between outside and inside, notice there's a lot more stuff out here than here. You still need a molecule to help get it from the outside to the inside. It can't just go on its own. It's facilitated by the presence of a protein. So it uses transport proteins, and that's what this figure is showing, facilitated diffusion. It's passive. What does that mean? Is it doesn't require energy from the body to do this. It's using just the concentration gradient where there's more on one side than the other, and that drives the process. I may ask you to describe this figure for the example. All right, primary active transport. Now notice the word here is active. This is going to require energy from you to do this. It uses ATP, which we talked about last time, adenosine triphosphate, to move products, and often it's going to move it in the direction it doesn't want to go as a result of diffusion. So it's going to go up the opposite. So it goes against the concentration gradient from low to high, which is the opposite of the way that it wants to go. So you're basically forcing it to go uphill instead of downhill, which is the way it wants to go. This is how nerve cells become ready to transfer information, how they can do the action potential, how your nerves become ready to have sodium potassium flow as part of the action potential. They move sodium potassium against their gradient using energy. So that's part of the reason why just thinking takes energy, is because as you use your nerve cells, you're pumping sodium potassium and it costs energy. Okay, this shows you how sodium potassium are moved. Now, I'm going to ask you on the test potentially to tell me about this figure. So we're going to start here, and I'm going to give you details, so take detailed notes. Now notice there's very little sodium here. These are little hexagons, these little stop sign shaped things, or are they octagons? 1, 2, 3, 4, 5, 6 hexagons. Okay, so these, what's going to happen is sodium comes in and it binds to the inside of this transport protein. Notice that there's room for 3. What happens next is we go to this next spot over here. What happens is when ATP binds to it, it causes the transport protein, instead of being faced inward, now it's faced outward. See, this says extracellular space. When ATP is used up, it ejects the sodium from inside to outside. So now we have even more going to where there is already plenty. Now, that's the energizing step of it. That pushes the sodium out. The next step is that potassium, which is shown as ovals, binds to the inside of the membrane. Notice the inside has, this side has these curved shapes. These receptors are curved shape that allows the potassium to stick. Now, as soon as the ADP leaves, that causes it to go from open to the outside to open to the inside again. And notice now that there's no longer these little divots there. And that pushes the sodium, or excuse me, the potassium to the inside where there's already plenty. And it causes the phosphate to be released, and it repeats. So it goes from sodium binding to ATP attaching, which causes the conformational change, switching from outside to inside. Sodium is popped out. The next step is potassium comes in. When the ADP leaves, leaving the phosphate there, that causes the potassium, which was bound there. Conformational change happens again. Now it's open to the inside again, and the potassium is pushed out. Notice that now there's lots of potassium in here, and the phosphate comes out. This ADP and phosphate eventually is going to make its way back to the mitochondria to be made into ATP again. So this is the cycle. Read about it in your text. I've gone through it in a little bit of detail. I may ask you to tell me how this works. Okay, phagocytosis and endocytosis. Phagocytosis means cell eating. Pinocytosis means moving small particles across a membrane. So phagocytosis is cell eating, and cells engulf large particles or whole cells forming a vacuum. Now this happens with white blood cells, this happens with amoeba and other individual cells that are going to be swallowing very large objects like another cell. So what happens next? Well, the lysosomes fuse with the food vacuole, which becomes what is called a phagosome. That dumps the digestive enzymes that were in the lysosome into the vacuole, forming a phagosome that digests it, breaks up that cell or organelle or whatever, and then the nutrients are absorbed into the cell. Now, this is how amoeba get nutrition, but this is how your white blood cells destroy bacterial cells or viruses that they happen to come across. In endocytosis, it's different. Instead of bringing a whole cell in, you're trying to take a small molecule by forming a vesicle around it. And this shows you how it works. Now notice that the star fits in with this little green receptor. And what happens is invaginations or infolding happens, and what was once on the outside is now on the inside of this little bubble and is transported somewhere to be used. And this is how you can get a molecule like this star-shaped molecule inside so your body can use it. In this case, you don't need a receptor. You're just going to have molecules coming in, the vesicle forms. Most likely, though, you have the molecule, the ligand stick to the receptor, invagination using a molecule called clathrine, and now you have the material that you want inside. That's endocytosis, moving something inside of the cell. It can be receptor mediated. Notice this one is receptor mediated, but this one is not. Exocytosis uses the opposite steps. There would be something in here, it would fuse with the surface and then be dumped to the outside. So basically imagine bringing it in and then dumping it to the outside. That's exocytosis. Okay.